Hello, and thank you for joining us. I'm Gwen Taylor, Senior Editor with Current Protocols at John Wiley & Sons, and I'm delighted to introduce today's webinar titled A Translational Proteomics Workflow Coupling Immunoenrichment to Selected Reaction Monitoring Mass Spectrometry and its Application to Alzheimer's Disease. This webinar is being co-sponsored by Current Protocols and ThermoFisher Scientific. ThermoFisher Scientific is a world leader in serving science, providing analytical technologies, reagents, consumables, services, and software for cutting-edge scientific research to routine industrial applications. Current Protocols is the largest collection of peer-reviewed, authoritative, and regularly updated step-by-step research techniques and procedures available for life scientists worldwide. With 17 titles and over 15,000 protocols, Current Protocols is part of Wiley Publishers. We have allotted 1 hour for today's program, but our speaker has agreed to stay on a bit longer to answer as many of your questions as possible. You can submit your questions throughout the event by clicking on the Q&A box at the bottom of your screen. The webinar will be recorded and available for viewing in the next few days. We will send each of you an email with details of how to access the recorded webinar, along with a PDF of the slides. Now it is my pleasure to introduce today's speaker. Dr. Mary Lopez is the director of the ThermoFisher Scientific Biomarker Research Initiatives in Mass Spectrometry, or BRIMS, Center in Cambridge, Massachusetts, where she is in charge of developing comprehensive mass spec-based workflows spanning sample preparation to bioinformatic analysis for robust biomarker discovery and clinical verification. Dr. Lopez has also held positions as Strategic Collaborations Leader and Proteomics Business Leader at PerkinElmer Life and Analytical Sciences, and as Executive Vice President of Proteomics Research and Development at Proteome Systems. Dr. Lopez has published extensively in the proteomics field and serves on the Scientific Advisory Board of the Clinical Proteomics Research Center at Massachusetts General Hospital. So, let's begin with a very warm welcome to you, Dr. Lopez.
Thank you very much, Glenn. And it's my pleasure to talk about some science that's very close to our hearts here at the BRIMS Center. Today I'm going to talk about the development of multiplexed Mesa, or Mass Spectrometric Immunoassay, SRM mass spec methods for proteins that are associated with Alzheimer's disease and its application to plasma samples. So I'm going to begin by talking a little bit about the proteins that we have developed the methods for in this particular study. And I'll begin by talking a little bit in general about Alzheimer's disease. A number of proteins, including amyloid beta, apolipoprotein E, and ceruloplasmin, have all been implicated in Alzheimer's disease, or AD. The leading theory of AD pathophysiology is the amyloid cascade hypothesis, which invokes the extracellular deposition of beta amyloid peptides, or A-beta, in large fibrillar aggregates in the brain. To date, several truncated variants of A-beta, amino acid 1 to 38, 1 to 40, and 1 to 42, have been found in brain and cerebrospinal fluid, while the data from blood and plasma are much more ambiguous. Apolipoprotein E is thought to play a central role in the AD amyloid cascade, and it is also associated with cognitive decline. Apo E binds to A-beta and both promotes A-beta fibril formation in the brain and also promotes A-beta clearance from the blood. These apparently contradictory fibril promotion and clearance roles for Apo E might be due to the different binding capacities of Apo E isoforms, Apo E2, 3, and 4 for A-beta. A third protein that we have developed methods to measure is called ceruloplasmin, and it has been associated with the Alzheimer's disease process because aluminum may cause altered synthesis of proteins that mediate copper homeostasis. And the proteins that would be affected would be proteins such as ceruloplasmin and ATPase 7B. The resultant excess free copper is associated with Alzheimer's disease, and so measurement of ceruloplasmin could potentially lead to an indication of the state. I'll talk a little more about the amyloid cascade hypothesis. The amyloid cascade hypothesis posits that the deposition of the amyloid beta peptide in the brain parenchyma is a crucial step that ultimately leads to Alzheimer's disease. Presenilin protease cleaves intact APP to produce fragments of A beta. The most common fragments that have been associated with Alzheimer's disease are A beta 1 to 42, 1 to 40, and 1 to 38 amino acids. I'll also talk about how apolipoprotein E is associated to AD. Apolipoprotein E is a protein that transports lipoproteins, fat-soluble vitamins, and cholesterol in the lymph system and then into the blood. Apo E is polymorphic with three major isoforms, Apo E2, cysteine 112, cysteine 158, Apo E3, cysteine 112, arginine 158, and Apo E4, arginine 112, and arginine 158. Although these allelic forms differ from each other by only one or two amino acids at positions 112 and 158, these differences alter the Apo E structure and its function and have profound physiological consequences. The Apo E4 variant, in particular, is the largest known genetic risk factor for late-onset sporadic Alzheimer's disease. While the exact mechanism of how the Apo E4 allele causes such dramatic effects remains to be fully determined, evidence has been presented suggesting that it interacts with amyloid. And in the little figure at the top of this diagram, you can see how the Apo E4 particles and other variants interact with A beta fragments and are deposited to potentially lead to Alzheimer's disease. Another interesting point to note is that the Apo E allele frequency in the general population worldwide is, as shown in this table, the E3 allele, which is the most common and non-pathogenic form of Apo E, is in 77.9% of the population worldwide. The Apo E2 mutation is in 8.4% of the population worldwide, and that isoform is associated with cardiovascular pathologies. I won't talk a lot about or at all about that isoform today. The Apo E4 isoform is in 13.7% of the worldwide population in general, and that allele, especially in the form of E4, is associated very strongly with Alzheimer's disease. There are other mutations that can cause the different isoforms of Apo E2 and Apo E4, but they are in much smaller percentages of the population. The three that I've described here are the overwhelmingly the most common. So BRIMS collaborates with the Bleno lab at the Sahlgrenska Institute in Sweden to develop SRM methods for the detection of A beta in serum and plasma. It's clear that there is a need to measure A beta accurately in cerebrospinal fluid and plasma and serum in order to understand better the progression of Alzheimer's disease. The Sahlgrenska laboratory has previously published extensively on Alzheimer's disease and A beta, and they published a method for A beta quantification in cerebrospinal fluid. That publication is actually shown here. The reference is actually shown here. However, that method does not have the requisite sensitivity to detect A beta in plasma. So the goal of our collaboration was to increase the sensitivity of the method and to apply it specifically to measure A beta Apo E isoforms and ceruloplasmin in CSF and plasma. Now, in order to reach the lower levels of sensitivity that are needed for a method that is able to be applied to plasma, a new type of sample preparation method called Mesa, or Mass Spectrometric Immunoassay, was applied. Sample preparation using Mesa can provide almost 100x enrichment of lower abundance analytes for the development of either SRM-based assays or other types of methods. The workflow itself is automated, it's high throughput, it's sensitive, it's very selective, and it's scalable. In addition, this method can be multiplexed. And in the diagram on the right, you can see the actual workflow that we've developed. Starting with a sample of blood or plasma, the sample is extracted on a small pipette tip that has an embedded microcolumn of a material that will bind the selected analyte if the material has been activated with a specific antibody. During the sample extraction and the elution on the pipette tips, which is done in an automated manner, the sample is repetitively drawn up and through the microcolumn and then eluted into a 96-well plate. The extraction and the elution will take approximately 2 hours, and this, of course, depends on the affinity of the antibody for the analyte. Once the eluent is dispensed into the microtiter plate and neutralized, it will be washed and reduced, alkylated, and digested with trypsin if the method requires detection of tryptic peptides. In the case of A-beta, because we're looking at peptides that are not non-tryptic, that are actually native and endogenous in the blood, we would not do this step. The reduction, alkylation, and digestion with trypsin takes approximately 3 hours. For the detection of Apo E peptides and ceruloplasmin peptides, we would actually apply the reduction, alkylation, and digestion with trypsin. Once the samples have been processed using this method, we can then go directly to the mass spectrometer and do the SRM assay, where using a fast flow chromatography, each assay or each method for each peptide takes approximately 1.5 to 7 minutes per sample. And in each of these sample runs, we can, again, by using a multiplexed method, measure all of the peptides simultaneously. This is an extremely powerful method that allows us to measure a large quantity of peptides in a particular sample at a high level of sensitivity. One of the qualities of the Mesa technology is that it is much more selective than other immunoenrichment techniques, such as the use of magnetic beads. For example, in this image that you can see below, a high-resolution LCMS of IGF-1 peptide that was enriched from plasma using the Mesa technology was compared to the same process using magnetic beads. And as you can see in the top panel versus the bottom panel, the Mesa-enriched sample demonstrates highly improved signal-to-noise. There are very few interfering peaks located around the actual IGF-1 peptide peak, as opposed to the result when magnetic beads were used, where there was quite a bit of nonspecific binding and some non-IGF-1 peaks that were also injected into the mass spectrometer. This, of course, can cause problems when quantification of the particular peptide that one is interested in and trying to measure is important. So in this slide, we're showing the complete selection of proteins and peptides that were targeted in this study for the development of AD-related protein Mesa SRM methods. And a total of 22 peptides were actually measured in the assay, and these consist of intact fragments of A-beta, 1 to 38, 1 to 40, and 1 to 42. And the amino acid sequences of all the peptides are shown, again, in this diagram. For Apo E, we monitored a large number of peptides. The collection of peptides shown here are actually Apo E monitoring peptides, and they're common to all of the Apo E isoforms, A3, A2, and A4. We also monitored a collection of apolipoprotein E isoform-specific peptides. These peptides are found only in the isoforms that are indicated on the right. So if you look closely, you can see that there are a number of Apo E4 specific peptides, which would only be found, again, in samples that contained that particular isoform. The last peptide in that sequence, which is amino acid 1 to 30 C to R, or cysteine to arginine, is the common mutation that is found in the Apo E4 allele. That is the mutation that has been associated most prominently with Alzheimer's disease. And the last selection of peptides are monitoring peptides for ceruloplasmin. In this slide, you can see some examples of data that were acquired from the samples that we measured, and it gives you an identification or a view of what the actual data from these types of experiments look like. So in the top left panel, we're looking at a calibration curve from A-beta 1 to 40. And in this curve, we can see that we have very nice linearity. It's the R squared is approximately 0.95. And if we go to the table on the right, we can see that we're actually measuring the peptide in the range that is the clinical target range, which is 20 to 80 picograms per milliliter. Our lower limit of quantification in this particular method was 50 picograms per milliliter. In the panel on the top right, you can see an extracted ion chromatogram from the A-beta amino acid 1 to 42 peptide. And what I want to point out in this particular chromatogram is that the peak shape is classically a very good peak shape, and that there's also very little to no other interfering peptides that are seen in this chromatogram. That allows us to very accurately quantify the amount of this peptide in the sample. In the lower panels, you can see similar types of data for Apo E and for ceruloplasmin peptides. And again, in these calibration curves, we can see that the linearity is very good. Again, R squared of 0.93. And again, in the extracted ion chromatograms for these peptides, we see a very good peak shape and a very great signal-to-noise and lack of interfering peptides, so that quantification can be done in a very robust manner. In this next slide, I'm going to present the data on quantification of A-beta peptides in the human cerebrospinal fluid that we used for the assay. So, as is consistent with previously published data, the Mesa SRM method was able to detect all 3 A-beta fragments in cerebrospinal fluid from individuals with AD. And here in this graph, you can see the relative amounts of each of these 3 fragments. The A-beta 1 to 40 was detected in the highest amount in the cerebrospinal fluid. In this next slide, I'm presenting data from the detection and quantification of A-beta, Apo E, and ceruloplasmin in human plasma samples. So for our study, we tested 4 pools, each of AD and non-AD plasma samples. And when the Mesa SRM methods were applied to this plasma sample set, a very different abundance pattern was evident between AD and non-AD samples. The levels of A-beta 1 to 42 were quantitatively increased very clearly in AD versus non-AD samples. And this would be consistent with our expectation that these fragments would be present in higher amounts in AD samples. Now, although we did detect A-beta 1 to 40, the signal was not quantifiable and below the level of ability to quantify, and the A-beta 1 to 38 was either absent or below the level of detection in these samples. The overall Apo E abundance of all the isoforms, as determined, again, by the average of all the monitoring peptides, was increased in the non-AD samples in this case, and that is shown pretty clearly in the graph. And the ceruloplasmin abundance, again, an average of all the monitoring peptides, was not significantly different between AD and non-AD samples. In this slide, we are presenting the detection of the isoform-specific Apo E peptides in the AD and non-AD samples. So as mentioned in some of the previous slides, we monitored 4 isoform-specific Apo E peptides, and these were all detected in the AD samples. The 2 unique tryptic peptides that were resulting from rare mutations were at a very low frequency in our sample set, and that would be these peptides that are shown with the double asterisks. These have actually been found in only a very few cases that have been published. However, peptide LGAD-MEDVR, which is the most common mutation and unique tryptic peptide for Apo E4, was found in approximately 1.4 times higher frequency in the AD samples. And you can see that illustrated here in the red box. Again, this mutation is found in approximately 13.7% of the population worldwide, and it is specifically associated with AD. So these results were very interesting and also confirmed many of the indications that have been presented in the literature with respect to apolipoprotein E4 and AD. The collection of Mesa SRM methods that have actually been developed at the BRIMS recently, including A-beta, methods for A-beta, Apo E, and ceruloplasmin, were recently published in this paper, and it details rapid development of sensitive, high-throughput, quantitative, and highly selective mass spectrometric and targeted immunoassays for a collection of analytes that are found in human plasma and serum. And the analytes that are described and the methods that are described in this paper are listed here. And there are 16 different analytes that are associated with a number of pathologies. So these methods are available and published. In conclusion, we can say that we have developed novel, multiplexed, and high-throughput Mesa or mass spectrometric immunoassay SRM methods for 3 AD-associated proteins. These methods demonstrate excellent linearity, sensitivity, and precision for A-beta 1 to 38, 1 to 40, and 1 to 42 peptides, and for ceruloplasmin and Apo E tryptic peptides. We've tested the methods with a small collection of AD and non-AD plasma samples, and we have demonstrated differential abundance of A-beta 1 to 42 and Apo E in Alzheimer's versus non-Alzheimer's samples. This is the first time that detection of A-beta has been shown in plasma using mass spectrometry. We've also demonstrated detection of Apo E-specific isoforms in blood plasma using mass spectrometry, and this is the first time that that has also been demonstrated using mass spectrometry. One of the Apo E isoforms, unique to Apo E4 but not to Apo E3 or Apo E2, displayed a higher frequency in the AD versus non-AD samples. Unlike previously published results, we did not observe differential expression or abundance of ceruloplasmin in our sample set. This concludes the data that I would like to present to you today in this webinar, and I thank you very much for your attention.
All right, thank you, Mary. Before we proceed to the question and answer session, I'd like to invite you to complete a very short survey immediately following this webinar. Excuse me, and in appreciation for completing the survey, you will be able to have a free download of a current protocols article on preparation of proteins and peptides for mass spectrometry analysis in a bottom-up proteomics workflow. And I'll give you more information on how to access that survey right after the Q&A session. So let's go ahead to the Q&A segment. And if you haven't yet submitted a question, now is the time to do so by clicking on the Ask a Question box at the bottom of your screen. So let's see what questions have come in so far. All right, the first one here I see is, does the Mesa protocol require a robot?
Hi, this is Mary Lopez, and the Mesa protocol does not require a robot. In fact, there are two different ways that the protocol can be applied. One is using, for very high throughput, one is using a robot that can handle 96-volt plates. But for experiments with small numbers of samples, an automated pipetter can be applied to the technology. So there's quite a bit of flexibility for this protocol, and it certainly is scalable.
All right, next question here is, what type of chromatography was used? Was it nanoflow or something else?
So the chromatography was actually fast flow, and one of the goals of developing this method was making a method, again, that could be done in high throughput and applied to large numbers of samples. So the chromatography was actually fast flow at a rate of approximately 200 microliters a minute, and that meant that each one of the samples could be measured in somewhere around 1 to 2 minutes.
Okay. Next question is, how were the target peptide sequences chosen?
So we chose the target peptide sequences using a couple of different methods. Primarily for A-beta, of course, we were using the sequences that are found in the endogenous peptides. So those sequences for 1 to 38, 1 to 40, and 1 to 42 were determined by the sequences that are present in those endogenous peptides. And those are the specific peptides that we were wanted to detect. For the other proteins, Apo E and ceruloplasmin, we used tryptic peptides, and we prioritized the peptides in the following manner. For Apo E, we wanted to be sure that we chose peptides that were unique for the different isoforms. So, for instance, for the Apo E4 peptide, the particular sequence that I showed is unique for that peptide, and that was included in the assay. We also included sequences that were unique for Apo E2, and then we included a variety of tryptic peptide sequences that are found in the Apo E3 isoform, the most common, and so that we had a collection of peptides that were considered sort of monitoring peptides for E3, and then other sequences that were unique for Apo E4 and Apo E2. For ceruloplasmin, we actually started with all of the tryptic peptides that are present when the protein is digested with trypsin, and then prioritized the peptides based upon their signal-to-noise in the assay. For all of the SRM methods that we do, there is a sort of a prioritization and optimization that happens when we are creating the assay or the method, where we test the signal-to-noise of each peptide and then focus our efforts on the ones that give us the best signal-to-noise or the ones that are absolutely necessary for measuring unique isoforms.
All right. Next question is, has the Mesa method been applied to other diseases yet?
Yes, absolutely. And in the paper, the clinical biochemistry paper that I showed in the next to the last slide, that was published last year, there are actually 16 different proteins that are related to a variety of different pathologies, things like sepsis, etc. And all of these were developed by using the Mesa protocol. So we are currently increasing our menu of assays and continuing to develop more as we address other types of pathologies or diseases.
All right, next question. What kind of antibody have you used to immunoprecipitate amyloid beta?
So we're using a commercially available antibody, and the details for where you can buy that antibody, where that can be purchased, are, again, in that clinical biochemistry paper, so I encourage you to take a look at that. In fact, there are a number of commercially available antibodies for A-beta, and we are actually in the process now of looking at all of those and trying to find the ones that give us, again, the best sort of signal-to-noise and have the highest affinity for A-beta. And it will vary depending on, again, the isoform that you're trying to look at, whether it's 1 to 38, 1 to 42, 1 to 40, or other shorter fragments. Something to note is that the antibody that we did use was bound all three of those forms, so it was not specific for an individual sequence, but it bound all three sequences of the endogenous peptides.
All right. Excuse me, next question. How does Mesa compare to, it's an acronym, says SISCAPA, SISCAPA?
Yes. So the fundamental difference between the Mesa approach and the SISCAPA approach is subtle. So both of these methods utilize antibody enrichment of a particular analyte and then delivery to the mass spectrometer. In the SISCAPA protocol, specifically, the antibodies are to peptides. And so when the method is applied to a sample, tryptic digestion happens first, and then the antibody enrichment is done to that fully digested sample to pull out specific peptides. So for the SISCAPA protocol, an individual antibody to a tryptic peptide is needed. For the Mesa protocol, although there is no a priori reason why we could not bind antibodies to peptides on the Mesa microcolumn, typically we do not do that. We use an antibody that is to the protein, at the protein level, to pull down or to bind the protein, and then that protein is subsequently digested with trypsin and presented to the mass spectrometer. The reasoning for this is that we're very interested in looking at different isoforms of proteins, similar to, or such as I showed with the Apo E. And if we use an antibody that is a pan antibody and will bind all of those isoforms, we can then pull down the collection of isoforms and individually characterize them on the mass spectrometer.
Okay, next question. It says the 1 to 42 data from AD versus non-AD are not entirely convincing. Is there a statistically significant difference?
Yes, that's a great point, and I do agree with you on that point. The sample set that we used for this particular study or this demonstration was very small. It consisted basically of a 20-sample 20 actual donor sample set, which we combined into 4 pools. And the reason we did this was because we were, again, stretching the limits of sensitivity of the assay. We, of course, will plan to apply this to much larger sample sets, and potentially the statistics will be better when we have more samples or larger cohorts. The second point that I'd like to make to that is that most of the data that have been published on A-beta have been in CSF and cerebrospinal fluid, and the levels of A-beta in plasma are much lower, and the differences between AD and non-AD samples may be more subtle than what is typically seen in cerebrospinal fluid. So we're actually, I think, breaking a little bit of new ground here, and the purpose really of this study was to first develop the method and then have the capability to apply it to larger sample sets. And we expect that our colleagues and our collaborators at the Solgrenska Institute will be doing a lot of that work.
All right. Next question. It says, you showed that the lower level of quantification was in the range of 100 picograms per mil. Was that in plasma or CSF, and what is the volume of plasma or CSF used for the Mesa SRM assay?
Great questions. So the LLOQ, yes, was in the range of about 100 picograms per mil, actually closer to 50, and that is in the clinical range, and it was measured in plasma. So that's calculated in plasma. And what was the second part of that question?
Saying what's that in plasma or CSF, and what volume of plasma or CSF do you use?
The volume that we use is a mil of plasma, and that's actually a wonderful question because it points out one of the sort of advantages of this Mesa protocol. Because we're using a microcolumn in a pipette tip, and we're actually drawing the sample up and down through the tip, through the column, multiple times to bind and elute, we have a lot of flexibility in the volume of sample that we can use. So for very high abundance proteins, like, for instance, Apo E or other apolipoproteins, we can start with a very small volume of plasma. Several microliters would be plenty in order to be able to have enough to measure in the mass spec. In the case of something like A-beta, which is present in much lower amounts in plasma, and we're really stretching the limits of these assays to try and measure those very low amounts, we can, with the Mesa protocol, we have the flexibility to use a larger volume. 1 mil in this case, we could have used 2 to 3, even 5 mils if we needed to, and then optimize the assay by drawing up and down through the tip, of course, in an automated fashion to get optimal binding and elution.
Okay. What was the lowest LLOQ you detected by SRM, and what's the most sensitive instrument, in your opinion, for targeted proteomics?
Oh, great. These are really excellent questions. So the lowest LLOQ varies, obviously, with the protein and the signal-to-noise, and all of those qualities or parameters will affect that. As an example, I will use parathyroid hormone, which is one of the analytes that we measured in the clinical biochemistry publication. And in the case of that particular analyte, it is present in very low amounts in plasma, and we're able to detect and quantify levels in the 8 to 10 picogram mil per range. So we are able to get down pretty low for some of these analytes. For A-beta, again, it was in the 50 to 100 picogram per mil, and so on. And in my opinion, which, I think it's only my opinion, actually the triple quad mass spec, which we used to do these SRM assays, is quite sensitive and can give us extremely good selectivity. I think that's sort of the best feature of this instrument. and it is extremely well-suited to measuring peptides and small molecules. However, there are other instruments which use high-resolution accurate mass detection, such as the Q-Exactive or the Orbitrap. And those instruments are also very well suited to looking at very low abundance analytes, especially if enrichment is done up front. One of the things that we're exploring in our laboratory is trying to measure some of these analytes in an intact form without doing any tryptic digestion, which would, of course, save an extra step in the whole process. And we have been able to reach pretty low levels in the picogram per mil range, looking at some intact molecules, things like insulin and so on, using those instruments. So I think it's a toss-up. It really depends on what you're trying to measure and how you're trying to measure it, which instrument is the best one.
All right, next question is, how did you reduce the binding of other proteins interacting with the target in the Mesa experiment?
Great question again. I showed in one of the slides the IGF-1 example. The properties of the Mesa microcolumn are unique in the fact that they are this particular type of matrix was developed specifically for mass spectrometry, and therefore the choice of the material and how the material was is put into the microcolumn and into the pipette tip is specifically designed to reduce to increase selectivity and reduce binding of other molecules that are undesired, so that when we apply the Mesa technology to a variety of different analytes, we see, such as I showed in the IGF-1 example, that we have very good selectivity and that the eluates are extremely clean, so that we don't have binding of other interfering molecules. And this is a problem, I think, that has been inherent with beads, magnetic beads, for many years. Researchers who are doing pull-downs often struggle with that problem. Another feature that we have implemented in our assays with the Mesa tips is to actually use a negative control where we bind an antibody that is not present in human plasma, such as beta-lactoglobulin, for instance, to the tip, and we do identical binding experiments in the same samples with that activated tip, and so we can see which proteins are binding non-selectively and which ones are binding selectively. It gives us a better ability to discern specific binding versus non-specific binding.
Okay, I think we can take a couple more questions here. One of them says, the data you showed also indicated Apo E3 related to a cardiac condition. Do you know if this study was extended?
We are currently investigating the distribution of Apo E isoforms, including E2, E3, and E4, with relation to mainly stroke and cerebrovascular conditions. There are a lot of literature references related to the Apo E isoforms and a variety of different pathologies, including cardiovascular, obviously cerebrovascular, stroke, AD, Down syndrome, and so on. Apo E appears to be a protein that has multiple functions, and so it is extremely interesting to follow up these connections between the Apo E isoforms and cardiovascular or cerebrovascular disease. We are studying specifically stroke and working with Mass General Hospital and Dr. Ming Ning on those experiments.
All right. I think we have one more question here. It says, would there be an advantage to using other enzymes such as lyse C or lyse N in the sample prep?
essentially, if you were interested in a particular peptide that was specific for an isoform or a condition, and you were not able to create that peptide using tryptic digestion, the trypsin didn't cleave in the right place, the peptides, tryptic peptides are too small for detection with a mass spectrometer, et cetera. Other enzymes, of course, can be used for digestion, so lyc and other types of enzymes for digestion could easily be coupled with this protocol.
All right. Thank you, Mary. Today's webinar has been recorded and will be available for viewing in the next few days, and we'll send each of you an email with details of how to access the recorded webinar, along with the PDF of the slides and instructions on how to customize and print a certificate of attendance. You can also access the certificate of attendance right now from the Resources tab at the bottom of your screen. Also in the Resources tab is the short survey that we're asking you to please complete, and when you finish that, you will get a free download of the Current Protocols article on sample prep for mass spec analysis. So this concludes today's webinar, and we look forward to your attendance at future events from Current Protocols. On behalf of our speaker, Mary Lopez, and from me, Gwen Taylor, and our sponsor, Thermo Scientific, we hope you've learned some valuable information today. Thank you so much for attending.